Good morning, students. In this video, I shall discuss the post-fertilization, its structures and event. Now, we have already discussed about the fertilization, how fertilization occurred in the flowering plant, and now, after fertilization, what are the different events which is actually going on there in the flowering plant. Now, after fertilization, these events are called post-fertilization event. And this post-fertilization event normally starts from the transformation of the different parts of the flower. Actually, the different parts of the flower, they are responsible for the formation of different parts after fertilization. But what about the different parts of the flower? What is their role after fertilization? We shall discuss here. Then after that, next one, that is the development of the endosperm, the reserve food for the developing embryo. Then after that, we shall discuss about the embryo, how the embryo develops inside the flower after the fertilization. Now, how the seeds develop from the ovule and the fruit finally develop from the whole ovary. So we shall discuss in detail about all these things. Now we shall start it with the transformation of the different parts of the flower. Now what are the flowering plants and how the different parts of the flower after fertilization they wither away and how they transform into different parts of the seeds and the fruits, etc. Now the different parts of the flower like your calyx, corolla, androecium and gynoecium. These are the four important parts. Now calyx consists of the number of sepals and the corolla consists of number of petals and the androecium consists of number of stamens, because the sepals sometimes they are totally wither away after fertilization. But in some plants like your brinjal, this part it is the calyx part, and this calyx part it is persistent. So sometimes the calyx may persist in some plants, but others it may wither away. But the petals mostly their main role is to attract the pollinators for the process of pollination. So that is why mostly the petals they wither away. And the stamens also, they actually prepare the pollen grains. After the preparation of the pollen grains, the anther dehisces. After that, the pollen grains are transferred to the stigma and their role is over. So that is why the stamen also wither away. But next, the style, stigma and ovary. These are the three important parts of the pistil. But the style and stigma, actually stigma, it receives the pollen grain from the anther. Then the style, it helps in the germination of the pollen tube and the movement of the pollen tube towards the ovule. But the ovary, now the ovary actually, it remains persistent and it transforms into different forms. Now here, the ovary, the whole ovary develops into the fruit. The fruit actually develops from the ovary. It is called the true fruit. Of course, sometimes the fruits are developed from other parts or other than the ovary. It is developing and they are called the false fruit, as in case of apple. Now, the ovule. The ovule actually develops into the seeds. The seeds which are formed there in the plant, and the seeds contains the embryo, seeds contains the cotyledon, then the seeds contains here endosperm, right? All these things. So here the ovule develop into the seeds. Now the integuments of the ovule, they develop the seed coat. The integuments means the outer covering. The outer covering of the ovule, they normally develop into the seed coat, and the seed coat protects the inner contents of the seed, the cotyledon, the embryo, etc. Now the next one, the outer and the inner integuments, they normally develop into the outer integument, develop into the testa, that is the outer covering of the seed, then the tegument, this is the inner covering of the seed. So the outer and the inner coverings, these are developed from the outer and the inner integuments of the ovule. Then the small opening, which is the micropyle, this micropyle now develops into the seed pore. Then the funicle of the ovule, it develops into the stalk of the seed. The seed attached with the part of the plant, that is called the stalk of the seed, and it is attached here. Then the nucellus layer of the ovule, which is present inside this ovule, it sometimes, this nucellus is consumed, it is utilized totally by the developing embryo, but sometimes it is persistent as the perisperm. When the nucellus layer, it is persistent, then this persistent nucleus is called the perisperm. Now, the egg cell, the most important part of the female flower, so that is the egg cell, which is actually developed into the zygote after fertilization, that is syngamy. During syngamy, the first pollen, it fuses with the egg cell to form the zygote, and later on this zygote develop into the embryo. Now the hilum of the ovule, it develops the scar of the seeds, and after that the synergid cells, they normally disintegrate. The synergid cells which are present adjacent to the egg cell on either side for supporting the egg cell, providing nutrition, and during fertilization the male nucleus enters through the synergid cell. So that is why the synergid cells after fertilization, they disintegrate and disappear. Also, the antipodal cells, the three antipodal cells which are present in the opposite side of the embryo sac, these antipodal cells, they also degenerate and disintegrate. So that is why this is the fate of the different parts of the flower just after fertilization. Now we shall discuss about the development of the endosperm. Now the endosperm actually develops from the primary endosperm nucleus, and this primary endosperm nucleus actually develops here by the process of triple fusion. During triple fusion, what happens? The second male nucleus or second pollen nucleus, it fuses with this diploid polar cell. Actually earlier it was the diploid polar cell having 2n number of chromosomes, because two haploid nuclei fuse together to form this diploid polar cell. It is in the central cell. Okay, this is the central cell, and this nucleus which is triploid in condition, and this primary endosperm nucleus, this is the primary endosperm nucleus, and this primary endosperm nucleus which is having 3n number of chromosomes, it divide and redivide, and the cell wall formation occurs first. The nuclear division occurs. After the nuclear division, then the primary endosperm cells are formed. So these cells are called the primary endosperm cells. Now the number of primary cells, primary endosperm cells increase here, and it fill up the full central cell and the nucleus, so nucellus. Now then these cells, they divide and redivide to increase their number side by side. The cell, or this is the embryonic developing embryo, you can say, and this embryo developed from the zygote. The zygote which was formed by the fusion of the first pollen with the egg cell, and this zygote, it divide and redivide to form the embryo, and this embryo develops from the zygote which is having 2n number of chromosomes, and one is divided 2, then 4, like that, but this cell, it is gradually developed into the embryo. And now these endosperm cells, they divide and redivide to form and fill up the entire cell, and this nucellus layer, you can say, and this is called a complete endosperm. And this complete endosperm, which is formed, precede to the developing embryo, formation of the embryo, because whenever the embryo starts development, it needs the nourishment or it needs the nutrients for its development, and all the nutrients normally supplied by the endosperm. So that is why the endosperm form first, then only the development of the embryo start, because it has to derive or it has to receive all the nutrients from the endosperm. Now, whatever seeds which are developed from the whole ovule, these seeds are mainly of two types, the endospermic seeds or non-endospermic seeds. These endospermic seeds means what? These are also called the albuminous seeds, where the endosperm is partly consumed and part of the endosperm remain persistent. So that is why these are called the albuminous or endospermic seeds. And the mature seed, normally it contains little bit of the endosperm. The endosperm is present in the seed. For example, in case of castor, sunflower, coconut, or rice and wheat. So these are some of the seeds where the endosperm is normally persistent or it is partly consumed by the developing embryo and part of the endosperm remain there in the seed. Now, in case of X-albuminous or non-endospermic seeds, normally the endosperm is completely consumed by the developing embryo. So due to which, so no endosperm is present in the mature seeds, but the cotyledons are only present, and these cotyledons normally supply the nutrition for the developing baby plant. Now these are the common examples like your gram, pea, groundnuts, bean. These are the dicot seeds normally, and two cotyledons are there, and they also support the developing embryo and the baby plant. Now, sometimes what happens, the remnants of the nucellus layer of the ovule also present, and these remnants of this nucellus layer, they develop into the perisperm. So this perisperm is special structure which is developed from the nucellus layer. So this is the development from the endosperm, and this development of the endosperm always precede the development of the embryo. Now we shall discuss how the embryo develops and receives the nutrients or nutrition from the endosperm. We shall discuss the development of the embryo, how the embryo actually develops from the zygote. So after the fertilization, what happens? Now the egg cell which is present here towards the micropylar end, the egg cell is now from the zygote, or the egg cell when fuses with the first pollen nucleus, it forms the zygote which is having in diploid condition. Now this zygote actually develops into the embryo, and the synergid cells which were there adjacent to the embryo, adjacent to the egg cell, they actually degenerate. They degenerate and disintegrate. Degenerate and they gradually disintegrate. But this is the zygote which actually develops the embryo. Now this zygote divide into two cells, after that 2 to 4, 8 like that. And the upper lowermost cell is this cell which is present in this part, that is the haustorial cell, which is the larger one. Then the next cell, or these are called the suspensor cell, and this part actually develops into the embryo. And this is the globular embryo, where the zygote divide into number of diploid cells. These are the globular embryo consists of number of diploid cells. Now after that, the shape of the embryo changes. Now these arterial cells remain here, the suspensor cells remain here, but the structure of the embryo changes into a heart-shaped embryo. And this shape of the embryo, now it is just like the heart of the human being, that is why it is called heart-shaped embryo. And after that, this embryo develops the radicle, which later on develop into the root, and now the plumule, which develop into the actual shoot of the plant. Now these are the cotyledons, which normally supply the food material or reserve food material to the developing baby plant. So these are the different parts which are developed from the zygote, and this is called the development of the embryo. Now after that, this embryo, actually it is remain in the seed. So in the seed, normally it is a dicot seed. So in this seed, you will find this embryo or developing embryo here in the seed. So in this seeds, normally you will find the cotyledons are there, and then the radicle and plumule, then the plumule, all are present here. So this is well protected inside the seed. So that is why these seeds actually contain the embryo. Then after the formation of the seed, then actually the embryo starts germination, and this germination, before germination, it undergoes the process of suspended animation, or you can say it is the dormant stage, and when it remains in the dormant stage, germination does not occur. But when the suitable conditions are available for the seed, then only the germination occurs. So now, after the development of the embryo, next the ovule of the female part of the flower, this ovule actually develop into the seed. And as we have already studied that the integument develop into the seed coat, then your micropyle develop into the seed pore, etc. So that is why the seed is developed from the ovule, and the whole ovary. Suppose this is the female part, and this is the stigma, this is the style that degenerate, but here this is the ovule. The ovule actually develops into the seed. This is the ovule, and inside the ovule the embryo sac is there, and inside the embryo sac this is the egg cell which develop into the zygote, then zygote to the embryo. But this whole ovary, which actually the whole ovary, it develops into the fruit, and this type of fruit which develops from the ovary, these are called the true fruits. These are the actually true fruit. But sometimes the fruits develop without fertilization. This is called parthenocarpic fruit. And sometimes the fruits develop from other part of the flower or other part of the plant. So that is called false fruit. In case of apple, there are some false fruit. And the fruits which are developed from the other parts, like apple, it is developed from the thalamus. So that is why this type of fruits are also observed in the flowering plant. So that is why the seeds develop from the ovule, and the whole ovary develops into the fruit. Now there are so many other mechanisms by which the fruit also develop, like the parthenocarpic fruits without fertilization they develop. So this type of fruits also occur in the flowering plant. Now I hope this video is useful for you, and if it is useful for you, then do like it and subscribe for further videos. Thank you.